Hey, good morning, everyone, and thank you for coming. It is quite an honor to be here, and so I'd like to thank the Society for supporting my work and inviting me to be here. And I'm very excited to share with you the fascinating chemosensory systems of bats. Pheromones are chemical cues that are excreted from individuals, and they mediate many social behaviors prevalent across all of mammals. So things like scent marking using urine or scent glands, identifying offspring or other conspecifics of the groups, or synchronizing your reproductive system or detecting if other members of the group are reproductively viable. Are you ready to mate? Are you not? Am I ready to mate when you are? And all these very kind of gross things that mammals do are mediated by a system called the vomeronasal organ. This is the system that is detecting pheromones. In the mammalian nose, there are two chemosensory systems. So highlighted in blue is the main olfactory system. And this is where a majority of the olfactory receptors are located that bind to many odorant compounds. But sitting anterior in the nose is an organ called the vomeronasal organ, or the VNO, as I'll call it. And this is the system that is binding to pheromones. And all these behaviors are very critical to mammalian fitness related to marking territory and reproduction. And so we would expect that the vomeronasal organ would be under strong selection and well conserved to maintain function. However, we do see some instances of loss across mammals. So for instance, in many aquatic mammals, such as cetaceans or sirenians, the aquatic environment has relaxed the selection and the vomeronasal organ has become vestigial, as well as in pinnipeds and the river otter. Interestingly, also in old world primates, including us humans, we do not have, as an adult, a functional vomeronasal organ. And the leading hypothesis is that as we have become more visual, selection has relaxed to maintain a well-developed chemosensory system as we need to respond more readily to visual cues. And finally, the other major pattern of loss is across bats. So most bats do not have a functioning vomeronasal organ, but the reason remains unexplained. So we're going to explore this question of whether vomeronasal function evolution is shaped by shifts in the environment, like we saw with the aquatic species, perhaps as bats evolved active flight, or a sensory exchange, such as using echolocation. So like I mentioned, the vomeronasal organ is mostly non-functional across bats, and here I'm showing you a histological section showing that the vomeronasal organ is quite small and rudimentary relative to the two groups in which there is a functional vomeronasal organ. So interestingly, there are two groups of bats that have a well-developed vomeronasal structure, the epithelial tissue and lumen of the organ here in Miniopterids is shown, as well as in the New World leaf-nosed bats. A group of neotropical bats that also have a lot of well-developed structures and epithelial tissue. So we need to answer a few questions before we answer our large one here. We have to find out when selection pressures change during bat evolution. Did these species regain vomeronasal function? And do these bats that actually do have a functioning VNO, are they using similar pheromone detecting mechanisms as other mammals? So if we map the distribution of vomeronasal organs across the families of bats, we see there are two instances in which the organ is functional, and there's basically two scenarios in which this could have evolved. So first and most parsimonious is that the ancestor of all bats had a non-functional VNO, and the system regained function twice. Alternatively, and far less parsimonious, is that the vomeronasal organ was functional in the ancestor and subsequently lost multiple times across the tree and has been retained in the few extant lineages that have it. Now, instead of using a morphological approach to look at this, I used a genetic approach looking at these two pieces of the genetic machinery of the pheromone detecting pathway. So a pheromone binds to a vomeronasal receptor called the V1R, and this signals a cascade that activates an ion channel called Trpc2, and this depolarizes the cell, sends the signal to the brain that recognizes the pheromone. So now to answer our first question, when did the selection pressures change in bat evolution, we're going to first look at this piece of the genetic machinery called Trpc2, the ion channel that has been described as indispensable to vomeronasal function. When Trpc2 gets knocked out in mice, all sorts of wacky social behaviors happen. The males are no longer able to recognize females from males, all sorts of weird mounting behaviors. The female no longer identifies her pups appropriately. So it's very, very tightly linked to social behavior and vomeronasal organ function. And when I say the Trpc2 is functional, I mean that the protein is intact, it's functioning appropriately. But I'll also be talking about pseudogenes here, and what I mean is that there is a mutation that has occurred in the gene that renders the protein non-functional. So there's either a stop codon that cuts it short during translation, or a mutation that causes the deletion or insertion that totally renders the protein non-functional. And in species where the VNO is vestigial, Trpc2 is a pseudogene. So across many of the aquatic and semi-aquatic mammals, as well as in the catarrhine primates, Trpc2 is a pseudogene, including in us humans. So I sequenced the Trpc2 in over 100 species of bats to try to get an idea of who has it and who doesn't. And I particularly focused on these groups that have a functional vomeronasal organ. And this is the tree of the species that I sampled. And interestingly, I found 13 independent pseudogenizing mutations of Trpc2 across the tree. This is 3 times the loss seen in any other group of mammal. So massive, widespread loss across the tree. And some of these losses, the mutations are shared among species, and some of these are happening more recently, not shared among other species. But in the groups where Trpc2 is functional, such as in the miniopterids, or where they have a functional vomeronasal organ, the Trpc2 is functional, as well as in noctilionoids, which includes the leaf-nosed bats, Trpc2 is mostly functional, with a few exceptions, such as in the Caribbean nectar feeders, which is a story for another day. But we see some more recent losses within phyllostomid as well. So now that we have a more clear picture of the pattern of losses across bats, we can answer our question, when did the selections shift? And in order to do this, I'm going to use a statistic in molecular evolution called omega. And this is the ratio of the rates of non-synonymous to synonymous substitutions. A non-synonymous mutation is one that changes the amino acid in the protein structure, where a synonymous mutation is a silent substitution that doesn't have any effect on the protein structure. And if a gene is under strong purifying selection to maintain function, we expect very low rates of omega, close to 0, as amino acid changing proteins are expected to be relatively rare because we don't want the protein to be non-functional or to be disrupted. However, as selection relaxes, non-synonymous mutations may accumulate and omega estimates will shift to the right, towards a more neutral signature of evolution where the 2 rates are in unity. So it no longer cares that it's accumulating non-synonymous substitutions and on its way to becoming a pseudogene. So we're going to estimate omega for 2 branch classes, the branches in which Trpc2 is non-functional and the branches in which Trpc2 is functional, as well as in the background branches. And we're going to simulate sequences that were under the same selection constraints as estimated from our Trpc2 1,000 times and see what sort of estimates that we get. And there are 2 scenarios I was expecting. So one, if Trpc2 had retained function, That means in the ancestor of bats, the Trpc2 was under purifying selection, and we expect a low distribution for those estimates, as opposed to a higher rate, more close to 1 for the pseudogenized lineages. However, if Trpc2 had reactivated, that means that Trpc2 was evolving neutrally in the ancestor and is indistinguishable from what we estimate from the extant lineages that have a pseudogene. And indeed, what we found was that omega for the functional branches was not only distinct and a separate distribution, but the distribution is very narrow and the variance is very low. And this is suggesting that it was not only under purifying selection, but invariantly so. So it has been functional for a long time, and it remains to be until today. Whereas the estimates of the pseudogenized branches have a much wider variance, suggesting that Trpc2 has lost function across different times and more recently. So when did selection pressures change during bat evolution? This is actually after most major bat families have diverged. It was actually the alternative and far less parsimonious scenario. Now, with these species that have maintained function through time, are they still detecting pheromones despite all these major masses of losses seen in other bats? Are they still detecting it similarly to other mammals? And we're going to focus on the superfamily noctilianoids, and this includes all of the New World leaf-nosed bats. And this is a fascinating example of adaptive radiation. This is a group of bats that is eating pretty much everything under the sun, including the vampire bats feeding on blood, a whole bunch of nectar feeders, a whole suite of frugivores, carnivorous bats, a really fascinating family to study diversity. And we're going to look at this other piece of the puzzle, the V1R receptors. These are the receptors in the gene families that the receptors are directly interacting with the pheromones. And so because they're responding to these diversity of chemical cues, the variation of V1Rs across mammals is highly variable. So we have orders of magnitude differences across different species. And what is known in bats at the time when this was published is that the two species that they had looked at, they found no functional V1R receptors. And we're going to turn to our friends, the primates, here to set up our hypotheses because they're showing similar patterns of losses, not quite to the extent that we see in bats, but they also show, for example, in mouse lemurs, diversification and the mouse lemurs having an order of magnitude higher levels of V1Rs. And what I'm showing you here is a gene tree of the V1Rs across mammals. And what I want you to notice is that the strepsirrhines, or the lemurs and lorises, are shown in blue, and they have some V1R receptors that are nested within other mammalian V1R receptors. So they share these receptors with other mammals. However, under that large blue arc is a duplication of a whole suite of genes that is only shared within the strepsirrhines. So they've had their own independent diversification of V1Rs. And so I think this is a good question to try to test for the noctilianoids, since they are also quite diverse. So is the bat VNO, like other Laurasiatherians, are they sharing receptors with their close relatives, believe it or not, but their close relatives? Or is it like are they showing the profile like a strepsirrhine? Have they had their own independent bat radiation of V1R receptors? And to do this, I sequenced the vomeronasal transcriptomes of this organ. So this is tissue-specific genes that are expressed. And I took this approach because we really didn't know how many receptors are found in the vomeronasal organ. And so this was a way to take all the genes at once and then pull out the ones that we were looking for. And this fieldwork and sequencing was supported by the grants and aid here from the Society. So thank you very much. And so I sequenced across 5 different lineages across noctilianoids. And I combined our V1R profiles with other Laurasiatherians, including the horse and the dog, as well as the pseudogene V1Rs of the bats that had a non-functional vomeronasal organ. Now what I'm showing you here is my gene tree, and the receptors shown in black are the receptors that we identified from the transcriptomes. So first note that we are not getting the same remarkable numbers as the mouse V1Rs. And when we consider the other Laurasiatherians, these V1Rs are nested right within the horse and the dog. So believe it or not, some of these receptors have remained largely unchanged and under strong purifying selection since a bat and a horse shared a common ancestor. So do bats share similar pheromone detecting mechanisms as other mammals? The answer is yes, and it's remained largely unchanged throughout the entire diversification of all of bats. Now let's go back to our big question now that we're a little more informed. Is bat vomeronasal function shaped by a shift in the environment, such as the evolution of powered flight? The answer is no, since most of vomeronasal loss has happened after most bat families diverged. And is there some sort of sensory exchange or trade-off with the evolution of echolocation? Also no, because the loss of function is decoupled with the evolution of echolocation. And in fact, many of the other very extreme adaptations that we see in bats, such as very specialized echolocation, UV vision, heat sensing, thermoperception, these bats have a very functional, well-developed vomeronasal organ. So what is going on? A lot of bats still are using chemical cues to detect or to mediate many social behaviors, but if they don't have a vomeronasal organ, what is going on? And they're living in these colonies by the thousands, sometimes millions. How are they identifying one another if it's not using the vomeronasal organ? And this reminds me of other similar social hierarchies and social systems that we see in other mammals that do not have a vomeronasal organ. And I'm just going to put forth a hypothesis to remind you that there are two chemosensory systems in the nose. And there's more and more evidence that the main olfactory system is also capable of detecting pheromones. So perhaps this redundancy, given that sensory systems are really expensive to maintain, selection might relax and on maintaining both systems that are doing the same thing. So with that, I would like to thank all of my funding, especially the Society, as well as members who helped me collect my field data and my sequencing. So thank you very much for your attention too.
Couple quick questions. Any questions for Laurel?
Yes.
Does this protein Trpc2, does it have other known functions?
No, so this form of the protein is only expressed in the vomeronasal neurons.
Yes.
The last point is that they're expensive to maintain. How do you know that?
So there have been some studies that have actually measured how much energy it takes to, from a neurobiological and biochemical standpoint. And if you think about the brain and like, for example, in most bats, a majority of the brain is devoted to echolocation or responding to auditory cues. So there might be some sort of trade-off, but I don't think it's necessarily as elegant as this echolocation sort of chemical cue trade-off.